Okay, so welcome back to this next video in which we are discussing pain and analgesic drugs. Okay, right, so I apologise for the abrupt ending to the previous video. Okay, so we're in the process of discussing the receptors which detect noxious heat. Okay, so we just discussed TRPV1, which detects temperatures greater than 45 degrees Celsius, and in response to those temperatures greater than 45 degrees Celsius, opens and allows in sodium ions as well as calcium ions into the cytoplasm of the cell from the extracellular fluid, and this causes that receptor potential which can then lead to action potentials being generated within the nociceptor. Okay, so we were just about to discuss another example of a receptor for noxious heat, and the next one we're going to discuss is TRPV2. Okay, so TRPV2, again, it's a homotetramer of 4 TRPV2 proteins. So the TRPV2 ion channel consists of 4 TRPV2 proteins stuck together to make the full tetramer, so to make a homotetramer. Okay, and the TRPV2 ion channels respond, i.e. they open in response to temperatures that are greater than 55 degrees Celsius, so higher temperature threshold for activation than TRPV1. Okay, and again, when they open, they're going to allow sodium ions and calcium ions into the cell to cause depolarization of the electrical potential difference across the cell membrane. That will constitute a receptor potential, and this can then activate the firing of action potentials along the primary nociceptive afferent that is connected to that nociceptor. Okay, so those are the 2 receptors that I want to mention for noxious heat, the TRPV1 ion channel and the TRPV2 ion channel. Okay, now we're going to turn our attention to noxious cold, so temperatures that are noxiously cold, okay, so badly cold. Okay, so these are also detected by TRP ion channels, and again the TRP ion channels are going to open in response to too cold temperatures. So the first one that I'm going to mention is the transient receptor potential M8, so it's in a different family now. Rather than being in the V family of TRPs, it's now in the TRPM family, and it's TRPM member 8. Okay, right, so TRPM8, again, it's a non-selective cation channel that is formed by producing a homotetramer of 4 TRPM8 proteins, and it opens in response to temperatures that are below 15 degrees Celsius. Okay, the other important thing to say, which is similar to this piece of information here, that TRPV1 is activated by capsaicin, TRPM8 is the target of the molecule menthol. Okay, so menthol is something that is within toothpaste, and it's what makes you feel cool. Okay, so it's the reason that when you brush your teeth, your mouth feels fresh and cool. Okay, so menthol is capable of activating TRPM8 channels, okay, causing them to open and allowing sodium and calcium ions into the cytoplasm of the cell, which then causes the receptor potential. Okay, now, you might be wondering, well, menthol, it doesn't feel painful when you brush your teeth. Okay, and that's because the menthol will be activating TRPM8 channels on non-pain neurons. Okay, so on other sensory neurons, which will just detect temperature but non-noxiously. Okay, they'll just detect cool temperatures, but they won't trigger that painful sensation. Okay, if you activated TRPM8 channels on nociceptor neurons with menthol, then yes, it would cause a painful stimulation. Okay, right. So the next TRP channel that I want to mention, which is also responding to noxious cold, is the TRPA1 ion channel, so the transient receptor potential A1 ion channel. So again, it's in a different family of TRP channels. It's in the TRPA family. It's family A, member 1. Okay, and again, it responds to too cool temperatures. Okay, so it responds to temperatures that are below 5 degrees Celsius. Okay, so when this channel is in temperatures of below 5 degrees Celsius, it will change in conformation, open, allow sodium and calcium ions into the cell, causing a receptor potential, and that will then generate action potentials in our nociceptor and that's nociception now complete. Okay, right, so those are the 4 trip channels that I want to mention that are involved in detecting noxious thermal stimuli, so extremes of temperature, so extreme hot temperatures and extreme cold temperatures here. Okay, so again, I'll reiterate that these will be present not only on the thermal nociceptors, which remember are those ones which are attached to a delta fiber primary nociceptive afferents and which detect only extreme temperatures, but they'll also be present on our polymodal nociceptors, which remember are attached to C fiber type primary nociceptive afferents and which are activated by loads of different noxious stimuli. Okay, right, so those are receptors for noxious temperatures. Okay, now what I want to turn my attention to is receptors for other noxious stimuli. Okay, and the next one that we're going to go on to is mechanical. Okay, so these receptors are going to be present within the mechanical nociceptors and also within the polymodal nociceptors. Okay, right. Now the first thing I should say about mechanical receptors is that we don't understand mechanical receptors nearly as well as we understand the thermal receptors. Okay, so much less is known about the mechanical receptors than is known about the thermal receptors. Okay, we don't really even know the identity of the ion channels which respond to intense mechanical stimulation. Okay, however, the 2 leading ion channels for ion channels that respond to mechanical stimulation are piezo1 and piezo2. Okay, so piezo1 and piezo2, which are separate ion channels that both are sodium ion channels when they open. Okay, so the idea is that potentially these 2 ion channels underlie this transduction of the nociceptive stimulus into a receptor potential, i.e. in the form of the sodium current coming into the cell. Okay, so they'll be present potentially, if they are the correct ion channels, they'll be present in the mechanical nociceptor free nerve endings, and they'll also be present within the polymodal nociceptor free nerve endings. And then when intense mechanical pressure is applied, these ion channels will open, they'll allow in that sodium current, and that will cause the depolarization of the electrical potential difference across the cell membrane called the receptor potential, and that can then lead to the action potential. Okay, but as I say, these 2 are still controversial, okay, as to whether they are the true ion channels responsible for transducing painful mechanical stimuli. Okay, right. Now what we're going to go on to is those which respond to chemical noxious stimuli. Okay, so we're going to go on to the chemical receptors, and these are going to be present on the polymodal nociceptors. Okay, so chemical receptors present on the polymodal nociceptors. Okay, so we're going to see receptors for both protons, okay, so receptors for low pH, and also we're going to see receptors for ATP. So remember when we were discussing the different types of nociceptors and what they responded to, we saw that the polymodal nociceptors did respond to chemical noxious stimuli. Okay, and 2 of the key chemical noxious stimuli that they were capable of responding to were low pH, which is too high proton concentration in the extracellular fluid, and also extracellularly located ATP, which is a cardinal sign of tissue damage, okay, because ATP should not be in the extracellular fluid. It's usually kept inside cells. So if you've got ATP floating around in the extracellular fluid, that's a giveaway sign that cells have basically burst open and released ATP into the extracellular fluid. Okay, right, so we now want to look at the receptors that are present on polymodal nociceptors, which remember are attached to the C fiber type primary nociceptive afferents that are going to transduce these chemical noxious stimuli into receptor potentials. Okay, so we'll start with ATP. So the chemical noxious stimulus we're going to study first is ATP, adenosine triphosphate, and this is going to activate an ion channel known as the P2X3 ion channel. Okay, so let me just explain what the structure then of a P2X3 ion channel is. So once again, I'll draw the membrane as two parallel lines. So this line here represents the inner leaflet of the phospholipid bilayer, and this line here, the top line, represents the outer leaflet of the phospholipid bilayer. And again, I'll show the ion channel as a cylinder here, sitting in the plasma membrane. Okay, now P2X3 ion channels are trimers, okay? In fact, all P2X ion channels are trimers. There's a great family of ion channels that respond to ATP known as P2X receptors. This is a specific P2X receptor, P2X3, that we're looking at. So all P2X receptors actually have this basic structure where they are trimers, like so. Okay, and basically if we take out a subunit from this P2X receptor, and I'll just label it up. This is a P2X receptor, which are a whole family of receptors which respond to ATP, and this is the generic structure of all of them. Okay, so we're going to pull out one of the subunits of this P2X receptor here and have a look at its membrane spanning topology. So exactly the same protocols we used when studying the TRP channels. Okay, so let's have a look at the membrane spanning topology of this subunit here. Basically, it's simpler than the TRP channel subunits. Its subunit structure is that it has a single membrane spanning alpha helix followed by a P-loop followed by another single membrane spanning alpha helix. So it only has two membrane spanning alpha helices, 1 and 2, and then again you have this P-loop which will line the pore. So every single one of the subunits will contribute a P-loop which will line the pore on the extracellular aspect of the pore. Okay, right, so this is a P2X receptor subunit, or just a P2X subunit here. Okay, right, now there's loads of different types of P2X subunits. One of the P2X subunits that humans have is called P2X number 3. Okay, and basically you can make then a P2X3 receptor or ion channel by putting 3 of these together, i.e. making a homotrimer. Okay, so the P2X3 receptor means a homotrimer of P2X3 proteins bound together to make a full ion channel that can respond to ATP. And this is an example of a ligand-gated ion channel, an LGIC for short, because the binding of the ATP is going to cause conformational changes in the P2X3 receptor, which will then open the pore and allow sodium ions specifically to move through it into the cytoplasm of the cell. Okay, so now let's just put all of that together. So the idea then is on these 3 nerve endings here of these polymodal nociceptors, you're going to have these P2X3 homotrimers here. Okay, like so. So this is a P2X3 receptor, or a homotrimer, whatever you want to call it. Okay, and then if ATP is present in the extracellular fluid, this cardinal giveaway sign that there has been tissue damage, what will happen is that ATP will bind to the P2X3 receptors, trigger a conformational change which will cause the P2X3 receptor to open, and then the P2X3 receptor will allow sodium ions to move from the extracellular fluid into the cytoplasm of the cell, and there's your receptor potential, and again that can contribute to an action potential as we've discussed more generally earlier. Okay, right, so the other chemical noxious stimulus that polymodal nociceptors are going to be able to respond to is low pH. Okay, now we all know that low pH means high proton concentration, which is one of those annoying realities, unfortunately, because of the nature of the way we defined pH as the negative, the log of the concentration of protons. Okay, so basically, what we're going to have on the surface of our polymodal nociceptors, so that they can respond to low pH extracellular fluid, is ligand-gated ion channels which are going to respond to protons, basically. The ligand in this case is going to be protons. And I'll just stress again, the ligand in the case of the P2X receptors was ATP, adenosine triphosphate here. Okay, right. In the case of these next ion channels that I'm going to discuss, it's going to be a proton. A single proton is going to be the ligand. Okay, so these next ion channels that we're going to discuss are known as ASICs. Okay, so what is an ASIC? Well, the abbreviation ASIC stands for acid, then the S is for sensing, and then the IC is for ion channel. So these are acid sensing ion channels. Okay, so they are going to open in response to acid. Okay, right, so let me discuss now the structure of an acid sensing ion channel. So in fact, they have a very similar structure to the P2X receptors. And in fact, the acid sensing ion channels and the P2X receptors are grouped together in a larger superfamily of ligand-gated ion channels known as the P2X-like ligand-gated ion channels. Okay, so basically within the enormous family of ligand-gated ion channels, one of the massive great superfamilies within that is the P2X-like ligand-gated ion channels, and in this family you have the P2X receptors and also the acid-sensing ion channels, the ASICs. Okay, they are key examples of this superfamily of ligand-gated ion channels. Okay, so why are the ASICs grouped with the P2X receptors? Well, it's because all of these pictures that I have drawn here apply equally well to acid-sensing ion channels. Again, they are trimers made up of 3 separate proteins joined together. And again, if you look at the structure, the membrane-spanning topology of an individual subunit of one of these acid-sensing ion channels, again, it looks like this. You have 2 membrane-spanning alpha helices with a p-loop in between the 2 membrane-spanning alpha helices. Okay, so all of that stuff that we've talked about there holds just as good for the ASICs, and that's why both of these are grouped into a superfamily, and the characteristics of ion channels in this superfamily are that they're trimers and that the subunits have this membrane-spanning topology. Okay, right, so basically there are different members of the ASIC family. Okay, so there are 4 different genes for subunits of the ASICs. Okay, so the 4 different genes are ASIC1, ASIC2, ASIC3, and ASIC4, and these all code for subunits of ASIC receptors. Okay, so they code for proteins that will have this membrane-spanning topology, which can be used to make one of the trimers. Okay, now, even though there are only 4 genes in humans for the subunits of ASICs, there are actually more than 4 different ASIC subunits, and that's because 2 of these genes have multiple splice variants. So the first gene, ASIC1, actually codes for 2 different proteins. It codes for ASIC1A and ASIC1B, and ASIC2 also codes for ASIC2A and ASIC2B, which gives us overall 4 different ASIC subunits, because there are 4 produced here, and then these 2 just produce 1 splice variant. Okay, so that gives us in total 6 different ASIC receptor subunits. Okay, so this phenomenon here is the phenomenon of 1 gene having 2 different splice variants here. Okay, right. So that means that you can build many different trimers if you want to build an ASIC, basically. Okay, so you can build both homotrimers where you put 3 identical ASIC receptor subunits together to make the full trimer, and you can also build heterotrimers where in fact you have different ASIC subunits coming together to make the full ASIC receptor. Okay, so the trimer consists of different ASIC receptor subunits. Okay, right, and many different ASICs are found within the polymodal nociceptors, basically. Okay, so that's the structure then of these things. So you can make many different actual ASIC receptors because of the fact that there are so many different types of subunits and there are different ways of combining them together to make a trimer. But the overall picture then here is that in the 3 nerve endings of these polymodal nociceptors, you're going to have the acid-sensing ion channels here. So here is an ASIC. And basically, if the proton concentration goes up in the extracellular fluid, what can happen is these protons combined to the ASIC channels, they can trigger a conformational change which causes the pore to open, and then again you get sodium ions moving from the extracellular fluid into the cytoplasm, and that constitutes the receptor potential generated by the A6. Okay, right. So that's then our discussion of the receptors which respond to noxious stimuli complete for now. There are more that we'll come across later. So for instance, receptors to things like bradykinin. Okay, bradykinin is actually capable of activating nociceptors. Okay, but one of its major roles is in sensitization of nociceptors, which we haven't discussed yet, but we will come on to. Okay, and in fact, the mechanism by which bradykinin actually activates the nociceptors is very poorly understood. In fact, we're not going to be able to say anything about that when we actually do come to inflammatory pain. So what I've told you now is pretty much the full picture with regards to receptors for noxious stimuli of what is understood at the moment. But we will discuss this further later on when we come on to inflammatory pain. Okay, so there are other chemical mediators which can cause activation of polymodal nociceptors as well. Okay, right. So the next thing I want to discuss is voltage-gated sodium channels, okay? Because this is actually going to become very important later on when we discuss neuropathic pain, and it's also got some interesting clinical correlates with it. Okay, so we discussed broadly in the previous video about how the action potential involves these voltage-gated sodium channels and voltage-gated potassium channels. Okay, so we're going to now look at the voltage-gated sodium channels in a bit more detail, and we're going to see that there are different types of voltage-gated sodium channels, and we're going to look particularly at which voltage-gated sodium channels are found in nociceptive neurons, okay, so the primary nociceptive afferents, okay, and then what we're going to see is certain conditions which result because of mutations in certain proteins which make up voltage-gated sodium channels. So we've just done the process of nociception itself now. What we're going to look in more detail at now is the action potential itself, and we're going to do this by looking at the voltage-gated sodium channels that are used by nociceptors. Okay, so let me just talk you through then the structure of a voltage-gated sodium channel. Okay, so once again, here is the plasma membrane, okay, and voltage-gated sodium channels consist of 2 subunits usually. They can consist of 3 subunits, but we'll keep this simple and just have 2. Okay, so the major subunit is what's known as the alpha subunit, and that's the one that actually forms the pore. And then there's an auxiliary subunit, an accessory subunit, which is called the beta subunit, which sits alongside the alpha subunit and can modulate its activity. For instance, it can modulate what electrical potential difference you need to get across the cell membrane in order to actually cause the opening of the alpha subunit. It can also modulate the conductance of the alpha subunit. So once the alpha subunit is actually open, how many sodium ions can actually move through there. Okay, right, so let me colour these 2 different subunits in. So here is the alpha subunit, the main pore-forming subunit of the voltage-gated sodium channel, here in red. Okay, and here is the accessory subunit, or the auxiliary subunit, which is the beta subunit. Okay, so let me label this up. This is what we would call the accessory, or you could call it the auxiliary subunit. Now, voltage-gated sodium channels, they usually just have 1 accessory or auxiliary beta subunit. Okay, however, sometimes you can have 2, so you could have 2 beta subunits bound to the alpha subunit. Okay, so that's when I was saying about how usually they're just made up of 2 separate proteins, but can be 3 proteins. That's what I meant there. Okay, so that's the accessory or the auxiliary subunit here. And then the alpha subunit, here in red, this is the pore-forming subunit. Okay, this is the subunit that actually has the pore moving down the middle. Okay, now, alpha subunits of voltage-gated sodium channels are big proteins, okay? It's a single protein that makes up this entire pore-forming domain. Contrast that to all the other ion channels that we've seen, the TRP channels, the P2X receptors, the A6, which were all multimeric. They all have multiple proteins assembling into the pore-forming structure. The voltage-gated sodium channels, it's literally just one protein that makes the entire pore-forming structure, basically. And this is the alpha subunit. So this is a massive, massive great subunit. And then it's got this little beta auxiliary subunit stuck on the side of it. Okay, now, the important point here is that there are many different alpha subunits of voltage-gated sodium channels. Okay, so in fact, in humans, there are 9 different alpha subunits of voltage-gated sodium channels. Okay, and these 9 different alpha subunits of voltage-gated sodium channels are called NAV for voltage-gated sodium channel, and then you put a 1, okay, and then you put point, and then whatever number it is. I've put a 1 there because this is the first one, but then it will go on. So for instance, the next alpha subunit would be called NAV 1.2, and after that NAV 1.3, and then so on, all the way up to NAV 1.9 to give us all 9 alpha subunits of voltage-gated sodium channels. So these are the names for the 9 different alpha subunits of voltage-gated sodium channels. Okay, so the question then is, the voltage-gated sodium channels that are in nociceptors, are they using a specific alpha subunit? Okay, or rather, let me take that sentence back. The alpha subunits that are within the primary nociceptive afferents with the nociceptors on their ends, are they using specific alpha subunits to build their voltage-gated sodium channels? And the answer is yes, kind of. The 3 main alpha subunits that they use are NAV 1.7, NAV 1.8, and NAV 1.9. Okay, so these are the main 3 that are found within the primary nociceptive afferents, basically. These are the 3 main alpha subunits used to build voltage-gated sodium channels. Okay. Right, now there are some interesting correlates to this that have clinical relevance. There are certain conditions which result because of having mutations in NAV 1.7 gene. Okay, so let me start by telling you about a condition known as erythromelalgia. Okay, so erythromelalgia. Okay, right. Not erythromelalgia, erythromelalgia. Okay, so erythromelalgia. Okay, so in this condition, erythromelalgia, you have a gain-of-function mutation in NAV 1.7. Okay, so this is a gain-of-function mutation in NAV 1.7. Now, what does this result in? Basically, this mutation results in the alpha subunit activating to open at far too low electrical potential differences across the cell membrane. So remember, the alpha subunit of voltage-gated sodium channels is supposed to activate to open at an electrical potential difference across the cell membrane of around negative 40 millivolts. That's the normal threshold potential. Basically, in erythromelalgia, what happens is the threshold potential goes down, okay, to something like negative 65 millivolts maybe. Okay, so the threshold potential becomes far too low, so that actually the alpha subunits are activated to open at normal resting membrane potential, basically, which means that you just spontaneously generate action potentials all the time, basically, whenever the electrical potential difference is at resting membrane potential. So basically, in this condition, you're continuously producing spontaneous action potentials in your nociceptors. Now, erythromelalgia, I should say, is an extremely complicated syndrome because you have NAV 1.7 not just in primary nociceptive afferents, but also in a huge number of other neurons, and all of those neurons are going to start firing action potentials spontaneously. So it's a hugely complicated condition, basically, with a huge number of different components that are caused by this dysfunction of neurons all over the place. Okay, right, but one of the key things that happens is that these primary nociceptive afferents start firing spontaneous action potentials because of this gain-of-function mutation in NAV 1.7, and this causes deep aching pains that are spontaneous. okay, because of the activation of the primary nociceptive afferents. Okay, right, the other thing that I should mention about this condition is that it's going to be an autosomal condition because the gene for NAV1.7, which incidentally is called the SCN9A, okay, so SC, S is for sodium, CN is for channel, then 9A, unfortunately it doesn't match up with the numbers here, but that is the gene for NAV1.7. Okay, this gene is on one of the autosomes, it's not on one of the sex chromosomes, okay, and this condition is going to be autosomal dominant. Okay, now what that means is that you only need one such mutation in one of the copies of the SCN9A gene in order to actually get this condition. Okay, so remember, if I draw your two chromosomes here, and I don't specifically know which chromosome SCN9A is on, but it will be on one of the autosomes, so one of the chromosomes from 1 to 22. If these are the two homologous chromosomes, you're going to have two genes for SCN9A, one here and one here. You only need the mutation in one in order to get the disease, and that reflects the fact that this is a gain of function mutation. The protein is gaining a new function, okay, rather than losing a function, and therefore you only need one of them to gain this function in order to actually get the disease, basically, and that's why it's autosomal dominant. Okay, now, conversely to this, there are also mutations in NAV1.7 which have been documented, okay, and I should stress these conditions are quite rare, okay, but there are also mutations in NAV1.7 which have been documented that are loss of function mutations which stop it conducting charge altogether, okay, or rather stop it conducting sodium ions altogether, okay, and if you get two loss of function mutations in your two SCN9A genes, okay, so if I put these here, if you lose function in both of them, okay, that means that you're going to have no functional SCN9A's gene product, i.e. no functional NAV1.7 anymore, okay. Now what that actually incredibly produces is complete insensitivity to pain. You cannot feel pain, okay, and indeed I think the person who was first found to actually have such a mutation was performing in certain countries by stabbing himself and walking on hot coals, okay, and because he couldn't feel any pain and he was entertaining crowds by doing this, okay, so it's an incredibly dangerous condition to have because if you don't feel pain, you start doing these silly behaviors, okay. So insensitivity to pain. Okay, complete insensitivity to pain indeed. Okay, right. So what that seems to tell us is that NAV1.7 is incredibly important in conducting the action potentials in these primary nociceptive afferents. Indeed, without it, you don't seem to have enough voltage-gated sodium channels anymore to have an action potential that's capable of propagating. Okay, now why might that be? Well, let's think about what would happen if you decrease the number of functional voltage-gated sodium channels in the membrane. Okay, what this simple picture here doesn't really stress is the fact that to get this sort of change here, it is not just one voltage-gated sodium channel that you need to open, it's loads of them in this little section of membrane, basically. You're not opening just one, it's a huge number of them. Okay, now, if this little area of membrane doesn't actually have a great enough density of voltage-gated sodium channels, then the actual changes in electrical potential difference that you're going to be able to generate will be much more feeble. It won't look like this anymore. And the problem is that that's not going to be able to conduct by saltatory conduction, because to conduct the action potential, you needed enough sodium to come in at this patch of membrane that enough would diffuse over to the neighbouring portion of membrane to cause depolarization up to the threshold potential. And if you don't get depolarization up to the threshold potential, then it won't conduct, basically. So it doesn't matter that you've got some funny activity occurring here, some little minuscule action potential. If it doesn't conduct, then it's not actually going to cause any signal to be transferred to the brain. Okay, so if you've got too little density of voltage-gated sodium channels here, it can mean that firstly the action potential is hugely distorted, it's miniature rather than large, and also what that means is it's not going to conduct because you're not going to get enough sodium coming in to adequately depolarize the electrical potential difference across the neighbouring portion of the membrane, which is essential in order for the action potential to conduct. And therefore you're not going to get any signals coming up your primary nociceptive afferents, and therefore you're not going to get any pain. Okay, right, so we will call it there for this video, and in the next video what we will turn our attention now to is how these primary nociceptive afferents actually enter the spinal cord, which secondary neurons they then synapse onto in the spinal cord, and we'll then look at the spinothalamic tract, which carries the signal up to the brain.